Okay, good, and thank you, everyone. Thank you, Rajiv, and all the organizers for the very nice invitation to come. And I'm glad I could actually make it finally this year. So I'll just talk briefly about all of what we've been doing, some of the work we've been doing at GBS, and just all the different ways we can apply this in the breeding program, and even extending this out to like seed certification and stuff like that. So just by way of introduction, if you're thinking about the breeding process, we have a lot of material, elite material, some unadapted, some exotic, and it goes through this really like funnel-like process. And this is many years, a lot of resources typically going into this type of process. And so we're always thinking about ways to make this more efficient and larger and faster using molecular information. So things like optimizing parent selection, optimize diversity and crossing, F2 enrichment type like marker-assisted selection approaches, genomic selection for whole genome, and you get to these more difficult traits like yield, and then things like marker-assisted backcross conversion. And in the end, we produce varieties and we want to make this go faster and more efficient. And so just to really oversimplify, but to think about the types of markers that we want and the objectives, you can think about single locus typing, or a whole genome profile. And so this is typically targeting known genes that we know have a large effect and we want to drive the real frequencies in the breeding program. On the other hand, this is also the whole genome, typically to look at population structures and relationships across all the material. And so just different types of applications for these. This is genomic selection and association mapping. But the big thing when you're a breeder, you think about cost. And so here, you typically think about cost per data point. Here, I tend to think more about cost per sample. So once you reach thousands or tens of thousands of markers, at least from the genomic selection perspective, additional markers are not that valuable relative to lowering per sample cost, which increases the population size, and you get a lot bigger gains that way. So this is the trend line shown earlier, and yeah, the sad thing here is it's leveled out for 2 years. But thinking about genotyping-by-sequencing or GBS, I always show this slide. And this is really driven by these amazing developments in sequencing. It turns out this is really good for wheat and other polyploids where we have contact sequence now rather than just assaying a single mutation. And then I think the big one, especially for diversity studies, but also for germ breeding and germplasm, is that we discover all the polymorphisms simultaneously. And the biggest one here again for breeding programs is that the per sample cost is pretty low. So the GBS approach that we take uses a reduced representation here, and this is restriction enzyme based. And so we primarily use the 2-enzyme combination, PST1 and MST1. And they're inexpensive because we put a lot of samples together, and then the sequencing cost is spread out across all of those samples. And so, still by way of overview, just to really simplify this whole process, going from genomic DNA, making libraries and then sequencing those, and then really streamline bioinformatics process. Here again, this is so we can move through a lot of material, make selections, and then discard the ones that are not selected. So now I'm just going to go through a bunch of different applications of how we've been using GBS. The first application we're mostly interested in is in genomic selection. So just highlight some of that. So the keys to genomic selection here again, this is probably familiar. It's starting and needing a training population, basically to train the model, and then that's applied on selection candidates. And so, in the flow of this, and so the real key here is because we're discarding 50 or up to 90% of the material after the genotype, is that we want it to be inexpensive. I just like to throw in here also though that we need accurate phenotypes. So the upper bound of prediction models is the heritability of the trait. And so if you have lousy phenotypic data, then you're going to get lousy predictions. And so this is our first study where we used GBS for genomic selection. And it was really just this question, is it a suitable market platform for GS? And so this was on some cement material from the Semi-Arid program. We had a pretty small set of lines. And the key here was that these were unreplicated field trials from Overground. So really high quality phenotypic data from these good replicated trials. And so we genotyped these with the typical GBS, generated about 35,000 markers after filtering, and then just applied a simple ridge regression. And so you can see here we're able to very well on these PCA, first and second PCA, discern some of the population structure. These are colored here by families. And then we obtain prediction accuracies for yield. This is grain yield under drought and grain yield under irrigated conditions. Prediction accuracies of 0.3 to 0.4 up to 0.5 in some of the trials. And so this is for a low heritability complex trait. This is pretty encouraging and good prediction accuracies. So to look at other traits, and here again for genomic selection, the real payoff is the more expensive and the more difficult that the phenotype becomes, the more valuable it is to get prediction with the genomic selection. And so a great one in heat is obviously heat quality, where we think about milling and baking quality parameters. And so this again, this study I highlight here is on cement bread wheat lines. And these again were from replicated yield testing. So we ran GBS on the same set of material, and then used just a cross-validation approach to get an idea of model prediction. And so this is a really busy slide here, but what I want to point out here is for all of these different quality parameters, including test rate, grain hardness, mixing time, low volume, had really good prediction accuracies in the range of 0.3 up to 0.6. And so, and just an example of two of those, this is a mixograph, and so you're actually milling the flour, and then you mix it there and look at the dough strength. So these traits are measured in minutes. So obviously not real high throughput, but we had really good prediction accuracy. Same thing with a loaf volume, so actually baking a small loaf of bread and then measuring the volume on that. So obviously here again, low throughput, expensive trait. And so if we can predict these kind of traits with any type of reasonable accuracy, I think this is really where there's going to be a huge advantage to apply genomic selection. The last study to highlight here on genomic selection, this is some stuff from the hard winter heat region in the central U.S. And this is what we call regional performance nurseries. So each of the breeders each year will contribute 5 to 8 entries, and then the total set will be about 30 entries. And so this is really analogous to the international nurseries that are coming from Semi. These sets of lines then are sent back out to all of the breeders and then grown at 40 to 50 locations each year. And so we have a lot of historic data. These were established in 1931. For this set, we went back to 1992. We genotyped all of these entries, and then we used the historic data on tens of thousands of plots and hundreds of thousands of observations. And so this set, just the southern sets of nurseries, is about 800 entries. And so now what we can do is we can really look at predicting the classifications across years. And if you're familiar with the climate in this region, is really highly variable from year to year and location. But what we came out with is here, this is on the bottom here, showing the prediction accuracies basically across the full set of material across all locations. And what we can do is, because these are breeding nurseries and oftentimes a few of the entries are repeated year after year, you can really get a baseline, which is what I'm showing here, a baseline of the phenotypic correlation from one year to the next. Okay, so that comes in at about 0.28, I think. And so for each one of these points here, you can see that 9 times out of 10, the genomic selection prediction is as good or much better than what we can actually obtain with the phenotype. And this here again is because the environments are so variable, and because also with genomic selection, we're leveraging information across many, many years of evaluation to start to iron out these differences. What then we can start to look at, again, is to start to look at genetic correlations across these environments. And so here's all of the locations shown with these points here, and then these lines and the colors, the width of the line and then the color are showing the number of years and the strength of the correlation across these locations. And so we can see, and this correlates well with breeding knowledge of the location. And a lot of these locations have very good correlation year to year. And then when we get to these drier, more extreme condition environments, the correlation really goes down year to year and location to location. So this also can really start to inform how we do multi-location testing and dividing sets of interest across locations. And so just where we're at on applying the genomic selection and some of the projects we have going on, this is a new project that we just kicked off over the last couple of months in partnership with Genomic and Cornell. And this is applying on a large-scale genomic selection in the Genomic graduate program. And so on this flow diagram, really looking at the generations of material coming through the program in Mexico. And then what we're looking at is taking and phenotyping these same entries across 4 locations in South Asia, 3 locations in India, and a site in Pakistan, targeted specifically for optimizing yield under heat stress. And so that's what we're showing here is just the different locations, and here they are on the map. And so what we'll be doing is there's about 1,000 entries that are going to each of these locations each year. The previous set of material, about 8,000 to 10,000 entries for genotyping in the Genomic program, and then at that point, we'll be developing these models and applying genomic prediction and selections at that point. So now I'm going to move on to some other ways we've been applying the GBS. Here again, just to kind of highlight a pretty versatile, flexible tool. So now just to think about characterizing genetic diversity. And so a little bit of background on this, thinking about in heat, we're talking here, going from wild species to early domesticates, there's a bottleneck there. And then in the intense bottom breeding process, there's been further bottleneck. And so what this looks like in heat, I think Bill gave a really great overview here, but heat is hexaploid heat that we call bread heat. We went through 2 rounds of whole genome hybridization. First to produce the tetraploid, and then with that hybridization, the tertiary to produce the hexaploid. And this hybridization event here, going from tetraploid to hexaploid, is an extreme bottleneck in the heat genome. And so what we can do is we can go and compare that to some of these rich genetic collections that we have. And so at Kansas State in the Wheat Genetics Resource Center, we have 500 some unique expressions of the tertiary species. We genotype this collection with GBS. And just also note that I'll show how these are physiologically classified as well. And so here's the distribution and the location collections of those expressions across Central Asia. And here's a nice picture of this kind of weedy species. And so what I'm showing here is the genotypes for all of these collections put onto the first and second PCA. And so you can see right away that there's pretty strong genetic differentiation into 2 previously known 2 subspecies, and then from that differentiation period, point out that this PCA explains like 56% of the variance. But what I'm showing in colors here is actually the physiological classification. And so you can see there's the different, what they call subspecies, but there's actually pretty clear classification application. And this wasn't a real big surprise because it turns out the morphological characteristics aren't actually that good at describing the different subspecies. What we're showing here now is the third and fourth PCA, and these are colored by location and collection. And so now we can also really see that we can do a good job also differentiate geographic origins and our evidence by genomic separation. And so what we want to do then is we want to put this back into the context of heat, the hexaploid genome, and the breeding material. And so now colored in green is the germplasm collection, and in heat here is about 600 entries from the Genomic breeding program. And so you can see here on the first and second PCA and the third and fourth that as is well known that there's very limited diversity relative to the wild species. So now to think about how can we take really informed approaches to integrate this diversity back into the breeding program. And so that's what we see here in the So now I'll move on and talk about some genetic mapping, and this is the GBS turns into a really easy, inexpensive way to develop some high-quality. One example here, this is on the synthetic product population, and showing here mapping stem mass resistance to two races of stem mass in this population. And you can see we can generate an extremely high density genetic map. And then from that, you can really start to map with high precision QTLs and genes across this population. And so here again, it really makes any type of mapping study very tractable and relatively inexpensive. The cool thing is that because, like I said earlier, you can discover SNPs at the same time you do the genotyping. This really enables moving into these wild species, moving into basically any species you want, and straight away developing some really nice genetic research. And so this is an example from Semi-Arid intermedium. It's a wild perennial grass species. It has some potential for domestication as a perennial grain crop. And here's only showing one set of the linkage groups. It's a genome pretty similar to wheat. It's 15, 16 gigabase genome, hexaploid, allohexaploid, but it's also outcast. So we developed an F2-like population from just one self individual. We could put together a really nice high-density genetic map. And so the key here is then we can really think about having tractable genetic resources for basically any species. And so what we can do then is we can take this information back to the breeding program and immediately start to apply genomic assisted breeding approaches. And here again, you can kind of do away then with this idea of orphan crops. At the same time, we have translocations and integressions from intermedium into wheat that cover valuable disease, virus, insect resistance genes. And so this genetic resource then gives us high-density markers on those chromosome lines to start doing chromosome engineering, fine map those genes, and then move them into small segments integration. So now I'll talk about some marker-assisted selection, and then this gets into that framework of having single markers relative to our whole genome profile. And so one thing we've been trying to work out now is what we call SNP-GBS. And so the idea here was to have an open, flexible platform where we can target single loci at known targets and do this basically independently of our whole genome profile. And the real key here is that we just use a really small percent, a small capacity of the total sequencing range, here again to make it cost effective, because when you're doing a few number of targets, you don't need a ton of sequences. That makes it cost effective, makes it high throughput, and it makes it flexible because now what we do is we use a single set of barcodes that are different from the sets of barcodes used for GBS. And so kind of like the flow of this whole thing looks like, because earlier I showed developing the GBS libraries here. These are digested adapters, and then they're amplified and fluid into a single library here. At the same time, on a completely different set of material, we can amplify known targets, and then with the N13 tails, we can append on a different set of barcodes. We can pool all of those into a separate tube, quantify those, and then we just add them in at 1%. And so what happens here is we can separate these back out after sequencing. On this side, we can generate something like 50,000 markers on 96 to 192 individuals. They're really low coverage, just like a 1.5 to 2x coverage at most. On the other side, using 1% of that, you have about 2 million leads. But if you're doing something like a target of 10 markers and 34 individuals, you can have like 500x coverage on those. So you can really start to eliminate missing data at the same time. So we tested this out on some genotyping. We made a typical GBS library, then we amplified 4 SNP loci separately and added those in at 1%. One thing we tried was we just converted markers simply by taking the selective base off of the end, and so that now rather than doing a competitive PCR, you're just sequencing the known SNP. And so that worked really nicely. What I'm showing here was one of the Caspar markers where you can see this is the number of reads. The read count, each one of these points being an individual. The read count for allele A and the read count for allele B. And so you can easily classify those as 300 zygotes here. These were more as dominant markers because they're picking up the same parallels in other genomes, but at the same time, you can easily classify these markers based on the read count. So now the final thing is just to talk about how we can even tie this into the seed certification process. So now we're just starting to routinely apply this technology into checking certified seed lots to make sure that they're the correct variety. And so we talk a lot about breeder seed and kind of in the breeding process. The thing is that all these important steps that this goes to foundation seed, it gets increased, and then it gets grown and produced on farms. And so at this step, there's opportunity, and through the certification process and through going through many different channels, there's opportunities for varieties to get switched, and a farmer could be receiving seed that is something that they did not order. It could be a variety that's unadapted or a variety that's not well suited. It's just mislabeled. And so what we're doing in the Kansas Foundation seed is we're starting to implement genotyping in every certified seed box that comes through. And then through that, we can check the varieties. And so here's just a nice little case study on that thing. And this was from one of the extension officers. There was a farmer that came in and said, hey, I purchased this seed. It doesn't look like what I thought it was. And so they sent us just these 2 samples. We genotyped them and checked them against each other. They were less than 5% homozygous, so that told us that they weren't like seed mixtures, but they were true line, true varieties, and they were only 74% identical at all the shared markers that were not missing data. And so that told us that, so really for any 2 random varieties out there, they're anywhere between 60 to 80%. And so we could tell that they're different varieties pretty easy. We have a huge set of reference panel between genotypes just as part of our routine breeding efforts. And so we could go in there really easily and say for sample 1, it was 98.7 identical to the variety 4, and the other one is 99+% identical to endurance. And these are well within our 1% error rate for GBS. So we can easily go in, check the variety, and then give that information back to the farmers and seed growers. So with that, just want to give all the acknowledgements. My research group, SNP-GBS, really runs the GBS pipeline, generating most of the material. Trevor Reif has done a lot of genomic selection modeling that I talked about today. And Tracy has worked on the intermediate wheat grass stuff. Also the Wheat Genomics Research Group, my group, along with the Bick and Joe's group, and Alan Fitz runs the wheat breeding program at Kansas State. The group at Cornell is instrumental in all of the initial stuff we did with genomic selection. Jeff Edelman, now at Wisconsin, works with us a lot on the genomic selection stuff. And then of course the group at Senate, which does all the amazing breeding work. Robbie and Suzanne, especially key in these big genomic selection. So with that, I'll stop. But I do need to acknowledge USAID, Gates Foundation, and the Food Alliance